Okay, so I guess I will begin. Nice afternoon, everyone. So this is going to be a presentation that it's a little bit different from most other presentations today, because it is a combination of biology and computer science, and more specifically, how we can use computer science in a slightly different domain, not the mainstream finance, telco, etc. application, but how we can use AI and new computer science techniques to interpret the human genome and to assist in clinical research and clinical practice. I will very briefly introduce our company because we're rather small. We're approximately 80 people. We're a fully remote company. And what we do is we are the creators of a suite of applications called VarSome. Our users are both research labs and hospitals. And what we do is we take samples from patients, both cancer patients or any other type of illness. We try to isolate the mutations in the DNA of the patient and then interpret the mutations of interest and annotate them with all relevant information that will assist in diagnosis. I will have to share a few info on biology background. Don't be afraid, I am not a biologist, I am a programmer. I am not going in anything weird you cannot understand. The basis of our work is DNA. You might remember from your high school biology years that DNA looks like this double helix, and it is the hereditary material in humans and almost all other organisms. So all our cells have DNA in our nucleus. What is more interesting to us is how DNA is formed. All information in DNA is stored as a code of 4 chemical bases. These are adenine, guanine, cytosine, and thymine. So basically, we have a long sequence of 3 billion bases, each base being one of those 4 letters. Think of it like an alphabet, and different combinations of this letter make up words and phrases, which in the end dictate how we look. The next concept that we need to understand is what is a gene. A gene basically is a DNA strand. So this is the basic unit of heredity, what defines that I have brown hair or how thick they are or whatever is encoded in one or many of my genes. The most important purpose of any gene is to act as instructions to make proteins. Proteins are basically what makes our cells work. Everything our organism does relies on the production of these proteins. A second function of genes is to decide how other genes express themselves. So maybe me here and one of the audience members have an identical gene, but something in my body switches it off while it is switched on in the other individual, so something different happens. We would expect that things are linear, so the most complex an organism, the more genes this organism has. This is not true, because genes are not the same size or combined in the same way. For example, a human, which is a fairly complex organism, has 3,000 megabases of DNA, but 20 to 25,000 of coding genes. A single grain of rice, which should be much simpler, has actually more genes than we do. So I will share a very first glimpse of how our tool looks. On the top row, where you see the gray bars, is one of the 2 strands of the DNA helix. And if you can see there the small orange area, this is what we open on the second row, where you can see how this sequence of bases looks in just this small, tiny part of our human genome. On the bottom row are the proteins that are transcribed based on the DNA sequence above. And finally, what is a genetic variation or a mutation? Because this is what is actually our work. So genetic variation is the difference in our DNA sequences that makes us individuals. It can be a variation in our germ cells, that is the sperm and egg. In this case, we are born with this trait, and it is also inherited. And this is how evolution happens. Or we can have a mutation in any other cell, what is called in biology a somatic cell, and this is associated with cancer. So we're interested in both. As you might imagine from looking at the series of letters on the DNA sequence, variants or mutations in the DNA are nothing more than changes in the sequence of letters. For example, let's say we have the word cat, we replace T for R, we get the word car. It means something else, or it might come up with a completely meaningless word. If we remove completely the letter A from car, we get something that means nothing. So you can have all possible problems you could encounter with missing or wrong text. Pieces of the code missing, pieces of the code being inverted. And this can happen on a very small scale, where even a single base is affected, or it can happen in bigger pieces of the genome. For example, it is even possible for a complete part of one gene to be translocated with a part of another gene. Naturally, the bigger the problem, the bigger the variation, the most likely is that this is not a benign variation. It's not something that affects my eye color, but it is something that might actually kill me. Although this is not always the case, because even very, very small variants can have a significant impact. So our job is, as you can imagine, as I said, we have 3 billion bases in our body. So imagine that anything can happen to these letters. Of course, after a point, you stop being a functional human. You cannot even be born. But still, changes can happen anywhere, and they can be combined. So for practical purposes, they are infinite. Some of them are well studied, and we have literature that is very well known for them. Others keep having research done. So we have publications and articles and databases come up literally every day. And others are not yet studied and might never be studied. So we might encounter them in only one patient. And there we need also to do some prediction. Based on similar changes, what might this mean? So this process is called variant annotation. On the top left, here on the blue, you can see a specific variation with a very weird encoded name that the biologists use. And we provide the basic information that is well known about this variant. We mine several databases, I think about 140-something at this point. Some are research databases, some are commercial databases. We integrate them, we combine them, we try to interpret the most important points, and then we filter them and display them to the user. A second source of information is information about genes or variants and their association to potential diseases that we find in publications. And we're going to focus the next few minutes on this topic, which is more straightforward to understand, actually. We download weekly or even daily publications that we mine from the web, either abstracts that are free or complete publications that sometimes we have access to, sometimes not. And we try to find in there keywords that are relevant to our work. Keywords relevant to gene, keywords relevant to variants, and how they're associated with proteins, how they're associated with illnesses, or even how they work together. So this means we have literally millions of publications, and we also get similar input occasionally from users. For example, a researcher might be studying something, and they point us to the direction of new research that we weren't aware of, so we add it. So as you can see on the left here, for example, for this variant, we have found 84 contributions from our users. That means 84 articles linked that indicate that this specific variant, which is a very well-known cancer variant, is indeed pathogenic. And we mine on our own, on the gray box below, 3,044 publications are associated with this specific variant. And we need to understand what this means and how they predict pathogenicity for this specific mutation. So this is the last part that I will show. I will hand over to my colleague to explain how this works. In this specific article, you can see here the abstract. The variant we're interested in is called BRAF V600E. It is a well-known pathogenic variant associated to cancer. So what we do is we process the abstract and we tag all references to variants. So we identify that this specific abstract relates also to this variant. We find also all associations to genes. You can see here the rest of the letters are other genes, codes for other genes. We identify that it is related to melanoma and to tumors, plus several proteins and other medical terms. We have specific dictionaries we use from the medical community. Christos, to you.
Hello. So its genetic analysis can contain millions of variants for each patient. And our platform needs to be able to highlight the most pathogenic, or inform, so that clinicians using our platform can understand which of these variants are linked to specific diseases, or whether, for example, a patient with a specific mutation would be a good fit for, let's say, treatment with a specific drug. Of course, AI can help in this regard by automatically extracting knowledge from published literature on genetics and annotating our variants almost in real time. So in order to achieve this, we fine-tuned a transformer model with publicly available datasets on the task of named entity recognition. We created a list of independent classifiers, in certain cases predicting the same types of tags. As you can see here, for example, we tag for genes, diseases, variants, etc. We have, for example, we have 3 classifiers predicting for genetic entities, and we later combine their output in order to increase our overall F1 score. The main challenges we faced were mainly related to entity disambiguation. For example, hepatitis B can refer to a virus or a disease, depending on the context. Naming conventions of genetic entities are also sometimes not respected. Generally, biologists are more likely to share a toothbrush than a naming convention. And this makes differentiating between proteins or genes quite hard. Differentiating humans from other species genes was also important and context-dependent. And finally, genes are normally recognized by symbols, and they can have synonyms or even deprecated synonyms, and these clash with one another, so disambiguating them is also context-dependent. So we had to be able to link the tags of the text with entries in our database. In order to achieve that, we built our own abbreviation detection model to help in this regard. We also performed some syntax analysis. For example, you can see a single tag, BRCA1/2, was expanded so we can match 2 genes in our database. And finally, for each tag, we generate a list of candidates, which we evaluate based on their similarity with the actual publication. In order to do that, we had to build a context for each possible entry that we can compare with the actual publication. And to achieve this, we constructed a knowledge base populated with multiple sources that we populated with definitions, synonyms, associated terms. And building this context allows us to pick the correct candidate very accurately. So to conclude, we currently predict for 15 types of tags at the moment. Our taggers run on an L4 GPU on Google Cloud, and they can tag about 2 million publications per day. And our F1 scores for the NER task approaches 90% at the moment. The results of our work will be available soon in our platform. Thank you.
Any questions, or was it too exotic to have questions? Yes?
I'm just wondering, that's a lot of data to comprehend. I mean, you build a software that parses all those researches and finds do the calculations?
Yes, we do actually something that is a little even more complex than this, because we get samples from different formats. We might perform even the identification of the variants, or they might come ready, identified by third-party software. So then we scan monthly 140-something, I think 44 databases. Some of them are huge. They are in completely different formats. Each organization has their own approach about how to do it. We have even data sources that we need days to download because of their size of terabytes. We have to have scripts to process them and unify them in a way that we can store them in our own system, in a way that is identified by our annotation and our classification software. And then we daily add whatever new comes in. So yes, it's a very complex system, and this is why also our cost usually relates to space so far. I mean, our cloud costs, and not to processing.
Would it be easier just to have let's say, a template that all scientists feel somehow?
Yes, it would be very nice, but scientists even you mean fill in with what?
Just fill the required information in a specific format that will be...
We already do that.
We do it, but in the end, what we need, our input cannot be dictated by us. So this is what is available. We're not an early stage startup. We have actual customers, I mean hospitals, labs, researchers, and it is often very hard to even communicate with different departments in the same hospital. It's a completely different mentality. They know nothing about computer science, and most of them really don't want to learn. They want a diagnosis, that's it. Anything else? Thank you.